There's a cartoon drawing I saw on Instagram one time where in the first panel it's an earth covered in wilderness and animals and forests. And on the planet is a couple of tents huddled around a fire with a fence surrounding them to protect them. The caption said, "This is a campsite." In the next few panels, buildings start to grow and the forest shrinks, and the fence stretches across the planet until it's a little circle. And in the little circle are animals and trees fenced up to preserve them. The caption said, "And this is a zoo." The point is, even though the end result of field science is that specimens and samples end up in labeled bags and put in the freezer, out on this trip we are still that campsite with a fence around to protect us. We are just house guests in a place that's older than the human race and is ultimately apathetic if our trip goes sideways on us. Episode 2, The Why, a storm chaser kind of science.
It's not that much different from, going to the zoo and watching the elephant to try to understand elephant biology.
Here to talk more about the zoo versus campsite is Dr. Drew Steen, head of the onboard biology team that came from USC.
The problem we have in the ocean is that most of the cells in the ocean do not want to grow in the lab.
Both the science crew and the ship's crew are sat around the cafeteria-style tables, surrounded by condiments and anti-slip placemats for dining in rough seats. But Drew stands at the front near a TV with a slideshow, and this mess hall is now an impromptu lecture hall, with a kitchen in the back that you might hear sometimes.
It turns out that in messy environments like mud, it's a lot easier to look at the DNA than the mRNA or the proteins. That's what we're focusing on. Then we can sort of get a list of the names of genes in a sample, and then we can add up those functions and try to take a guess at what those genes are doing. And so it's a little bit like trying to figure out like what kind of work a person does based on the tools that they have, right? Like this person is clearly a carpenter, this person is an electrician, this organism has a gene for sulfate reduction and for sugar metabolism, and a gene that produces antibiotics, and a gene that makes a little tail that allows bacteria to move around. So we guess that it's an organism that eats sugar and breathes sulfate and goes to antibiotic war with the other cells around it, and maybe has a tail so it can move around when it doesn't like where it is. And that would be a fairly common set of characteristics for a cell in the subsurface to have.
Yeah, we're declaring right now that Drew has a doctorate also in science analogies. Turns out it runs in the family because he's married to Karen Lloyd, a marine microbiologist, and here's a little bit of her TED Talk to explain the global scale importance of our Alaskan sampling expedition.
And scientists and engineers and entrepreneurs are working on methods to pull carbon dioxide out of these point sources so that they're not released into the atmosphere. And they need to put it somewhere. So for this reason, we need to keep studying places where this carbon might be stored, possibly in the subsurface, to know what's going to happen to it when it goes there.
So what exactly happens down there? Though the real estate of the ocean floor and below does not have north-facing windows or much natural light for that matter, it's still home to a native species that are the center of both Drew and Karen's research. These slow-growing yet long-living microbes are called Chemolitho autotrophs. Here's Karen again to break them down.
Which is a microbe that uses chemicals, chemo, from rocks, litho, to make food, autotroph. These chemolitho autotrophs take the energy that they get from these processes and use it to make food like plants do.
Karen continues to explain the absolutely and quite literally metal way these microbes are surviving, which makes me believe that life on and under the ocean floor is more about the Bear Grylls life than it is Steve Irwin.
The bare necessities of life will come to you.
Yet, Karen is connecting the farm-to-fork diet of the Chemolitho autotrophs as being a solution to its local ecosystem-wide food shortage. But another type of storm is also a part of the Chemolitho low and slow cowboy life. And rather than a competitor or a predator, Chemolitho Cowboys and its neighbors face unseen threats to their lifestyle, like salt in the soil of a field.
So what can happen here will control how carbon and nutrients and trace metals are either trapped and removed from the ocean or released back into the water. And so this can control things like how quickly climate change happens, how big of an ecosystem the ocean can support, if there are toxic things like mercury in the water that your fish is eating. The first place that I really started to ask these questions was within the Arctic.
Enter Dr. Ali Tesson, co-chief of the research expedition with Drew and head of the Kent State Geochemistry team. This Alaska trip is one of many field research projects in her lineup, and as you just heard, her work in studying biogeochemical cycling as an indicator of climate change takes her around the world. Let's hear more from her about chemistry as a major player of this expedition.
I've talked a lot about carbon, which is what we were aiming to investigate in a lot of these projects. But on my first expedition, I got really excited about iron. So we were doing trawls and we started pulling up these like huge fist-sized chunks of iron oxide minerals that were precipitating on rocks, they were precipitating in burrows, they were precipitating basically everywhere, which highlighted that it was a really kind of unique iron cycle that we were seeing in these locations. We care about that because iron, as well as manganese minerals, do a really good job of trapping chemicals within their kind of solid phases. So what the iron's doing will impact, again, things like mercury and arsenic, which we care about because they're toxic, nutrients, which fuel ecosystems, and then also organic carbon, which again plays a role in whether we see climate change get worse or slow down. We also tend to care about iron because it can be a biolimiting nutrient. What that means is that process of photosynthesis is actually controlled by how much iron is in the water column. It is the limiting nutrient in the region that I was studying. Also a spoiler, it is the biolimiting nutrient where we are right now.
Headlines of Alaska in the last few months have shown pictures of rivers turning orange, specifically satellite images of 75 rivers in the state turning rusty. And it's all connected to what Ali talks about here. Statewide permafrost melt that releases iron into waters like rivers, which then empty into the ocean. With that, Ali's geochemistry team brings a research question on board.
How is iron being stored and mobilized from Gulf of Alaska marginal settings? So asking a similar question to what I've done in the Eurasian Arctic, but within the Gulf of Alaska, trying to understand the role that sediments play in trapping or releasing iron, which again drives all of those other ocean chemical changes.
And the key to understanding those ocean chemical changes is knowing the global marine speed and direction, just like driving out of a roundabout or traffic circle, depending on where you're from. And we have an expert on that too. Not traffic circles, but Paleoceanography, a third and crucial piece to the puzzle that drives this expedition.
Has everybody heard of the global conveyor, the Great Ocean Conveyor? So the difference in the radiocarbon age of interior ocean waters is how we figured out that ocean circulation ventilates to the surface in a few places and deep waters are formed and move around the planet in this conveyor belt.
The reason why Mo's team is looking at carbon dating to begin with is to fill a long-standing knowledge gap in the field of paleoceanography. See, the Pacific Ocean was not thought to be a major agent in past climate change studies because it was poorly studied for a variety of reasons. One of Mo's reasons is that major oceanographic institutions were in the Atlantic region to begin with. But we now know that the Pacific Ocean is one of the largest heat sinks, meaning that it absorbs 80% of climate change caused by humans and has a huge heat capacity, so is able to take the heat without changing temperature itself. Kind of like a really patient coworker.
Chef Sydney, you got that right?
Yeah, sure.
The Pacific Ocean reorganizes that heat. So a lot of that heat goes in at low latitudes, but then these big circulation patterns and gyres move that heat up to the high latitudes, which is where that controls Southern Ocean and Northern Pacific paleoclimate.
Alright, now is the time to buckle up for a ride back in time, as most team research focuses on a chronological interstate that connects current-day Alaskan waters to the Pleistocene period, where the last ice sheets were recorded, so the last recent millions of years.
The Pleistocene Pacific, it was still massive. It still contained a huge amount of heat. That heat, we have every reason to believe, was rapidly reorganized. Plus, we had a cute little ice sheet. And we are on the margin of that paleo ice sheet right now. So the Cordilleran Glacial Complex, the last remnants of that are the Alaskan glaciers.
So now that most team nailed the where of the Pleistocene ice sheet, their big question to answer on this expedition is the when, and that's why they depend on modern-day carbon dating technology. Because, as mentioned before, the Pacific's paleoceanography is not historically as well researched as the Atlantic. But even when faced with a prehistoric question, most team has a plan to pull data from physical samples. And so, let's revisit our precious deep-sea sediment samples to meet the neighbors of those Chemolitho Cowboys.
Right, the Heinrich events. But we don't really know, because it's gone. So how do we reconstruct this ice sheet? One of the things that we can do is look at foraminifera. So foraminifera are these little protists. Some of you may have seen them in the microscope downstairs as we've been pulling up cores. So protists are a different class of life. They're not plants, they're not animals, they're their own thing. They're single-celled organisms, and happily for us, they make this shell out of calcium carbonate, CaCO3. That calcium carbonate, it's a gift that keeps on giving. Because an organism that is precipitating carbon and oxygen out of the water is also taking in isotopic information on those different isotopes as they exist in the water at the time that that organism grew. So then we can pick these shells out of the sediment, we can dissolve them, and we can look at the flavors of carbon and oxygen in them, and we can reconstruct things like the temperature and the salinity of the water column, things that trace back to the stability and the meltwater input from ice sheets.
So to recap, we have a game plan on why 3 different disciplines and investigators joined up on the hybrid engine ice cutter Sikuliak vessel in the Alaskan Gulf. Conveniently, they will be using the same sample type, subsurface sediment. And over the next 2 weeks, Drew, Allie, and Mo will need to huddle around ocean floor maps and discuss often how to best chop up the 2 weeks and hit the most sampling or coring sites to meet the interests of the 3 different labs. In the next episode.
When this is full of mud, we put it into the mud.
We learn how to gear up and get our hands dirty with information-rich samples.